Hello friends, today we are going to learn about biotechnology applications. This is the second lecture conducted by me uh, in my channel Life Science at the rate Life Science for you on youtube.com. So, what we will cover in this chapter? In this chapter, we will cover recombinant DNA technology, that is RDT. Its short form is RDT. It is it is nothing but recombinant. First in this we are going to learn the tools of recombinant DNA technology. Here are which tools we use for the purpose of recombinant DNA technology preparation while the preparation of recombinant DNA and this is the whole technology, that is it conducts cloning and transfer of DNA and how the whole process gets carried out, that we are going to learn in this lecture. So now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the tools. So we basically in this we are going to learn the restriction enzymes, polymerase enzymes, ligases, vectors, and host organisms. These are the key tools that are used in recombinant DNA technology. So what is restriction enzymes? Restriction enzymes nothing but enzymes that cut the DNA at particular locations. These are known as restriction enzymes because they are enzymes. They are the proteins which act as enzymes. Okay. Restriction enzymes main purpose is to cut the DNA at a particular location. Polymerase enzymes. Polymerase enzymes are responsible for uh, just to polymerize the DNA and to make its number of copies. And ligases. What ligases stand for then? Ligases are nothing but the DNA for enzymes which ligate the DNA. That is they just go and they ligate the DNA on one above the order. Wo, that is they just joins the DNA molecules. Vectors are nothing but the transfer. Vectors are nothing but vehicles mainly that used to transfer the DNA from its donor organism to its host organism. So these are nothing but vectors. Let's try to understand this in details. Uh, After this, we are going to learn this all in details. Okay. So first is nothing but restriction enzymes. In the year 1963, two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. In the year 1963, two enzymes which are restriction enzymes got discovered from bacteriophage which were which were isolated from Escherichia coli. Escherichia coli is nothing but bacteria that is present in our gut. Okay, this bacteria is present in our gut, and bacteriophage is nothing but the organism that is responsible for its engulfment. Escherichia coli, it just bacteriophage is a parasite that just joins the Escherichia coli and transfer this bacteriophage's DNA inside Escherichia coli, multiply there and and prepare its billions of copies, and it joins, and bacteriophage when its uh, stage get accomplished of total formation, then it bursts, it leaks from that DNA, Escherichia coli, that bursts the Escherichia coli and comes out of Escherichia coli. So one of these added methyl groups to DNA. So after in response after the isolation of these two restriction enzymes, one was added to the DNA. Methyl groups are added to the DNA. That is methyl transferase, while the other cut DNA. So other DNA, other enzyme was restriction enzyme was used to cut the DNA. The latter was called as restriction endonuclease. So inside the nucleus, it cuts the DNA. Nucleus means it is endonuclease means inside the DNA it cuts. Inside the nucleus, endo means inside and nuclease means which is used to cut the DNA molecules. So inside the DNA molecules, it cuts the DNA and forms two different strands. Therefore, it is called as a restriction endonuclease. The first restriction endonuclease Hind 3 whose functioning depends on a specific DNA nucleotide sequence was isolated and characterized 5 years later. So in 1963, the two DNA restriction enzyme mainly got discovered. After that, after its discovery, after 5 years, what happened? The restriction endonuclease Hind 2 was isolated. So here what the Hind 2 was isolated and characterized means how it works. Characterized means how it works. It got discovered. It was found that the Hind 2 cuts the DNA molecules at a particular point by recognizing a specific sequence of six base pairs. So what found after this study? So after this study, it got found that Hind 2 cuts the DNA when a particular sequence of six base pairs got present inside the DNA. When this six base pair of DNA gets come, that time this restriction enzyme go and bind to that DNA molecule and cut at a specific location. So the specific base sequence is known as recognition sequence. So where these six base pairs comes, it is called as a recognition sequence. Why it is called as a recognition sequence? Because this sequence was recognized by Hind 2, and after its recognition it cuts at a specific location. Therefore it is called as specific recognition sequence of Hind 2. After that, besides Hind 2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, of which recognize different recognition sequences. So besides Hind 2, means Hind 2 is nothing but one of the example. After that, today we know more than 900 restriction enzymes. So more than 900 restriction enzymes today are gone, or we know. In fact, we know a database. We have a database of more than 900 restriction enzymes. Database is nothing but collection of the data that we have isolated from 230 strains of bacteria. So where these restriction enzymes got isolated? From 230 strains of bacteria, each of which recognize the different sequence. So recognition sequence is nothing but what? The specific sequence of six base pair or more base pairs to which a particular um, enzyme binds and recognizes it. So it is nothing but recognition sequence. The the convention of the name this enzyme is the first letter comes from the genus and second two letters come from the species. So first letter come from the genus, H comes for its genus, and the two letters after which are coming comes from the specific prokaryotic cell from which it got isolated. Example is E. coli R1 comes from Escherichia coli, where Escherichia coli is nothing but microorganism and RY is nothing but its strain and 13 is nothing but its number. So Escherichia coli R1 means Escherichia coli RY13. So how it got nomenclated? E for Escherichia, CO means coli, R means RY, 13 means 1. So it got its name from its genus, species, and from where it got discovered, and its strain specificity number. It is its strain specific number RY13. Okay. After that, in E. coli, the letter R is derived from the strain, name of the strain. So R is nothing but what? It's the strain name. Roman numbers following the name of the indicate the order in which the enzymes were isolated from the strain of bacteria. So a Roman number follows after this strain number, and this indicate the order in which the enzyme was isolated from the strain of bacteria. So the order, that is the first, second, or third. This is nothing but the order. Order is nothing but anything that is arranged in specific way. So order in which the enzymes were isolated from the strain of bacteria. So bacterial strain from which it is isolated, and its order, the order, and its gene, its species, its strain, and its order, whole this four five things gives rise to the name of for the name of restriction enzyme, that is EcoR1, whose first letter is from its species, CO for from sorry, its H, E comes from its genus, Co comes from its species, and R comes from strain, and I comes from its I comes from its uh, order. Okay, so all this comes from EcoR1, that is Escherichia coli RY13. Okay, so sixth point is restriction enzymes belong to larger class of enzymes called as nucleases. Nucleases are nothing but what? That that goes inside the nucleus, that recognizes the specific sequences, and that cut on a particular DNA molecule. After this, there are two kinds, exonuclease and endonuclease. Exonuclease removes the nucleotide from the ends of the DNA, whereas endonuclease cut at specific position of the DNA. So exonuclease is nothing but the enzyme which are responsible for the cutting of the end of the DNA molecules, whereas the endonuclease where it goes inside the DNA template and cut at a specific position. Therefore, them called as endonucleases. After that, each restriction endonuclease in function by inspecting the length of DNA sequence. So here what happens? Restriction endonuclease functions as a functions by inspecting. What is used to do? It inspects the DNA. It finds a particular repeating sequences inside it that are called as a tandem repeats. And finding these tandem repeats, it cut on a particular specific sequence and at a new particular nuclease base pair. And once it finds its specific recognition sequence, uh, now we came to know that recognition sequence means a particular sequence which is responsible for recognition of enzymes. Means enzyme recognizes the particular sequence that are called as recognition sequence, and this restriction endonuclease finds or inspects the DNA and finds its recognition sequence, and where it cuts the DNA molecule and separates it in two strands of the double helix at which the point the sugar phosphate backbone. So sugar phosphate backbone is present inside the DNA, and on the sugar phosphate backbone it cuts by recognizing a particular specific restriction endonuclease site. After each restriction endonucleases a specific palindromic nucleotide sequences in the DNA. So what is palindromic stand for here? The palindromic means a DNA molecule finds a particular sequence of 6 to 7 to 8 or more base pairs, which is palindromic, means it is repeating foreground and from uh, its first base pair to last base pair in reverse order on the second strand from last base pair to first base pair. That is how. So look at this figure. Here what happens: action of restriction enzymes. The enzymes cut the both DNA strands at the same site. This is the vector DNA molecule. This is the foreign DNA molecule. Okay. Here what is happening? We are cutting these two both the strands with the help of restriction enzyme EcoR1. EcoR1, where what is it? Escherichia coli RY13 cuts the DNA between the bases G and A only when the sequence GAATTC is present inside the DNA molecule. So here what is happening? The vector DNA is also conducting has repeating palindromic sequence GAATTC. And why it is called as palindromic sequence? Because it is repeating itself like this: GAATTC, GAATTC. Here again in foreign DNA molecule, GAATTC, and its reverse GAATTC. So this set of 12 nucleotides which are bind to each other, and mainly six base pairs which are repeating foreground and in backbone are recognized by Escherichia coli. So therefore, this palindromic sequence is called as a recognition sequence. And on this recognition sequence, after getting this G and AATTC, the restriction enzyme EcoR1 cuts at in between G and A, sugar and phosphate backbone. It cuts, and after cutting, here also it cuts in between GAA. Okay. After cutting, what happens? It creates a sticky ends. So what is sticky ends? Basically, this is nothing but these are the sticky ends. Here again on foreign DNA molecule, this is sticky DNA molecule. When these two sticky DNA comes in contact with each other, it joins and the recombinant DNA forms. So what is here? So the vector DNA and foreign DNA molecule, after getting a particular sticky end, it comes together, and with the help of enzyme ligase it get joined. So therefore it is called as recombinant DNA, and this palindromic sequence is called as sticky ends. So after, so what is the properties of this palindromic sequence? So here we will get an example. There are group of letters from which the same word of the end, both forward and backward, gets repeated. So here Malayalam is repeated in the foreground in a forward sequence like MALAYALAM, and if we will reverse it again, then we will get the same sequence MALAYALAM. So Malayalam is nothing but we example of a palindromic sequence. As against a word palindrome where the same word is read in both directions, the palindrome in DNA is a sequence of base pairs that reads the same on the two strands when orientation of the reading is kept the same. For example, following sequence, the read reads the same in the two strands of 5 prime to 3 prime end. This is also true if I read 3 prime to 5 prime direction. See, this is 5 prime to 3 prime, it is 3 prime to 5 prime. And this is DNA molecule. It is nothing but palindromic sequence. And after getting this palindromic sequence, in between G and A, restriction endonuclease are echo R1 cuts here. Therefore, it is called as a recognition sequence. This is nothing but recognition sequence. And here is a cut. And after here, again in between GAATTC, G and A, here echo R1 cuts. Therefore, it forms a G2CTTAA sticky cut. So it is called as a sticky end. Restriction enzymes cut the strand of DNA a little away from the center of the palindromic sites, but between the same two base pairs on the opposite sides. So what restriction enzymes used to how restriction enzymes used to cut the DNA? So here it cuts the DNA in the palindromic sequence in between the two bases. G and A is nothing but here two bases, and again here G and A again in opposite side, and in between G and A, and here opposite side again in between G and A, the restriction enzyme cuts. So this leaves the single stranded position at the end. So here what happens after getting DNA is nothing but double stranded molecule inside its eco R1 host or vector. And foreign DNA contains two strands, that is one is from 5 prime to 3 prime, other is from 3 prime to 5 prime. So this is nothing but the orientation of DNA molecule. Okay. So here what happens? Overhanging stretches called as sticky ends. So this CTTAA after getting cut forms a single strand, and it is called as sticky end. These are named because they form the hydrogen bonds with their complementary counterparts. So here what happens? The G and C are bi- get bound to each other with three hydrogen bonds, and A and T, that is adenine and thymine, uh, uh, again form two bands. A and T again form two bands with each other and get linked. This T again form two bands with this A, T again with A, and G again with C, or C again with G, with three hydrogen bands or bonds. So here what happens? G this AA forms a complementary counterpart of TTA. Okay. So here again we will see what is the counterpart of C? G. What is the counterpart of A? T. Again, T's counterpart A, T's against T's counterpart A, A's counterpart T, and C's counterpart G. Okay. These sticky ends. Again, what happened? The single-stranded DNA molecule at the end are is eager to join with uh, other end of cut DNA molecule, which is sticky. Therefore, it is called as sticky ends. And these ends, when come together with the help of ligase, it got joined. Therefore, it is called as a sticky DNA joining or ligation. And DNA, an enzyme which forms or which conducts this ligation of DNA molecules is called as DNA ligase. This stickiness of the ends of facilitate the action of the enzyme DNA ligase. Restriction endonucleases use in genetic engineering to form recombinant molecules of DNA which are composed of DNA from different sources or genomes. Here, restriction endonucleases are used in genetic engineering. So basically, what is genetic engineering? Genetic engineering nothing but finding a host molecule, again finding a foreign molecule or vector DNA molecule, joining them or cloning them and transferring it to a particular microorganism or eukaryotic cell and multiplying it and forming number of copies for the production of large scale a particular product, or for the formation of particular product in large scale. This experiment got conducted, therefore it is called as genetic engineering. With the help of genetic engineering, a genetically modified organism got created, and genetically modified organism is nothing but the organism who is genetically modified or whose genetic sequence, that is DNA sequence, is altered. Therefore it is called as GMO or genetically modified organism. So here again, restriction endonucleases are used in genetic engineering to form recombinant. Recombinant means again recombinant, who is he is combination of two or more things, and recombinant means it is recombinated. The two DNA molecules got combinated or recombinated with the help of DNA ligase. It is called as recombinant DNA, which are composed of DNA from different sources or genomes. This is maybe either a genetic source, that is a gene source, or a genome source. Genome is nothing but haploid set of DNA molecule of a or chromosome of a DNA or restriction enzyme. What can I say? A host. Sorry. Here genome stands for a haploid set of host DNA. When cut by the same restriction enzyme, the resultant DNA fragment have the same kind of sticky ends. When a host vector DNA and foreign DNA are cut together with the same enzyme, that is EcoR1, it forms the same sticky ends. Therefore, it is very easy for a ligase to join them and form a complete DNA molecule who is recombinant. Therefore, in this topic, sticky ends are these are got joined to each other with the help of ligase. But what if E hind 3 is cut by a vector is cut by hind 3 and the foreign DNA is cut by EcoR1, then are they able to join? No, they can't be able to join because they are cut from different sources. That is the restriction endonuclease used by them are different. Therefore, there is a uh, what can I say? Condition. There is a condition that the vector DNA should get cut with the same restriction enzyme to which the foreign DNA molecules being cut. Therefore, it is very, very important to cut the restriction DNA or with the help of restriction endonuclease at a palindromic sequence of vector DNA and foreign DNA by the same restriction endonuclease to form same sticky ends, and this same sticky ends get ligated to each other with the help of DNA ligase and forms the recombinant DNA. So this is foreign DNA, this is vector DNA plasmid. Plasmid DNA is nothing but circular, and foreign DNA might be for circular or it might be a linear. What happens when particular DNA sequence of palindrome or palindromic sequence, who is called as recognition sequence, got found on foreign DNA and vector DNA? The specific restriction enzyme cut this at that particular site. Okay, so after cutting, it forms the sticky end. Here is a sticky end. Again, here is a sticky end. Here is sticky end, and here is sticky end. These four corners of a sticky end. This corner comes in contact with this, and this corner comes in contact with this. And when they sticky ends got contact in with each other, that time DNA ligase joins the foreign DNA molecules to the plasmid. This foreign DNA molecule got joined with the help of DNA ligase to form a recombinant DNA molecule, and this recombinant DNA molecule again is transferred to the cell, and when cell divides, it forms its clone because there is no genetic variation as it is asexual reproduction. Therefore, here what happens when this cell get divide, it forms two cells whose genetic makeup is ideal to each other or 100% alike. Therefore, they are called as a clone of each other or clone of host, and they again contain this plasmid DNA inside them. So they are called as a clone of each other. So in case of Agrobacterium tumefaciens infection, what happens? tDNA, Ti plasmid got recognized. After that recombinant DNA, Ti plasmid was formed, and this recombinant DNA again transferred to or injected inside of plant cell on a particular chromosomal location, and after getting introduced, these cells grow and forms a plant, and this plant is called as a recombinant plant or genetically engineered plant or GMO plant or genetically modified organism or genetically modified organisms plant species. So unless one cut the vector and source DNA with the same restriction enzyme, the recombinant DNA molecule cannot be created. As again, we are getting again the same point that when we form two sticky end of vector and foreign DNA molecule, and again this two sticky end should be with the help of recombinant restriction endonuclease should be same in case of vector DNA as well as foreign DNA. And when this vector DNA and foreign DNA got cut with the help of same restriction enzyme, then they form the sticky ends, and then only they get joined to each other. Here again condition is there that unless one cut the DNA and source DNA with the same restriction enzyme. Okay, same restriction enzyme is used in both the cases. Separation and isolation of DNA fragments. Our next point is separation and isolation of DNA fragments. The cut DNA by the cutting of DNA by restriction endonuclease result in the formation of fragments of DNA. When DNA cut, it forms a fragments. Therefore, it is called as fragmentation of DNA or fragments of DNA. These fragments can be separated with the technique called as the gel electrophoresis. Here, gel is nothing but a state of a neither solid or neither liquid. Gel is intermediate step, which is viscous, who is somewhat rigid, but not hard as solid. Therefore, it is called as a gel state. And electrophoresis is nothing but in this uh, electrophoresis is nothing but migration of DNA molecules or any molecule in with the help of anode, cathode, and inside the gel they got separated according to their size and shape. Therefore, with the help of electric pulse, we run DNA molecules and separate them. Therefore, it is called as electrophoresis. Since DNA fragments are negatively charged molecules, they can be separated by forcing them towards anode by moving them towards the anode under an electric field through a medium or matrix. So here, since the DNA fragments are negatively charged, so DNA contains negative charge on its phosphate group, that is PO4 group, and this negatively charged DNA molecule gets separated by forcing them towards allow them to move towards the anode. Anode is nothing but the anion, where anion moves. Anion is nothing but negatively charged, and cathode is nothing but positively negatively charged, and uh, cathode which moves towards the cathode. Therefore, they are called as cations, and cations are mainly positively charged. Therefore, here anode act as the receiver of or positive charged electrode, and electric field was applied, and medium is nothing but gel to separate the DNA molecules. So after that, nowadays the most commonly used matrix is agarose. Agarose is uh, came from the natural polymer extracted from seaweeds. A red algae or red seaweed is responsible for formation of agarose, and highly purified form of agarose or agar is called as agarose. And this highly purified agarose, when we boil it and cool it inside the water or buffer, it forms a gel-like semi-solid state. Therefore, it is called as a gel. And agarose is a natural polymer extracted from the seaweeds. The DNA fragments separated resolve according to their size through sieving effect. So, what is sieving effect here? Here, the DNA fragments separate or resolve according to their size. Either they are more big than larger size, which moves very slowly inside that matrix, that is gel, while the negatively charged short DNA fragments moves very slowly. Therefore, they are called as um, slow moving, fast moving molecules. The DNA fragments separate and resolve according to their size. Resolve means get separated according to their size through a sieving effect provided by agarose gel. Agarose gel, what here agarose gel form gel stand as a matrix, and it gives a sieving effect to resolve the DNA molecules or DNA fragments, or to get separated according to their size. Size is nothing but big or small. Hence, the smaller fragment of DNA again faster and more it moves. So again, hence the smaller the fragment size, the faster and more it moves. So DNA is 100 base pair long, then it will move very fast. If it is 200 base pair strong or long, it will move somewhat with less speed, 300 with very less speed, 400, 500 according to. So if DNA is big, then it will very slowly move, and if it is very short, then it will move very fast. Therefore, through the sieving effect of a gel, provide or gel is nothing but agarose gel, and agarose is nothing but the seaweed extracted from seaweed, natural polymer. So separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide. It is very important here that DNA which got separated, how it got visualized, because DNA can't we can't see DNA because DNA is very minute, and naked eye, with the naked eye we can't see the DNA molecule. Therefore, under the sieving effect of gel molecules, by moving them with the help of their size and shape, they get separated and stained by ethidium bromide. Ethidium bromide, it joins with DNA molecules, and it gives a red color, otherwise a color or white color inside the UV radiation. And this UV radiation got captured with the help of camera or through gel dog machine, and with the help of UV rays. That is, when these gel molecules come in contact with the ethidium bromide, and ethidium bromide after staining with ethidium bromide. You When we expose these DNA molecules to UV light, that is ultraviolet light. So after getting exposed to ultraviolet light, this ethidium bromide stained by DNA molecules, or stained with after these DNA molecules stained with ethidium bromide, form a red color, and this got captured with the help of camera, and we can see the clear picture of a gel. So the separation of a DNA are cut from the agarose gel and extracted from the piece known as the illusion. Illusion is nothing but the extraction. Here, DNA molecule got extracted from the agarose gel by cutting them at a according to their size or desiredness. This process is called as illusion. The DNA fragment purified in this way used in constructing recombinant DNA by joining them with cloning vectors. So how this recombinant DNA molecules got recognized or reconstructed with the help of recombinant DNA molecule by joining them with cloning vectors. So last point stands here. So how DNA fragments purified? So they are purified according to illusion. In the way are used to construct in recombinant DNA by joining them with cloning vector. So here what happens? Recombinant DNA again joined to cloning vectors or vectors, that is foreign DNA got joined with cloning vector with the help of sticky end after isolation from gel, cutting them. So therefore it is called as joining and cloning. So here a typical gel tank with proper supply. Here this is nothing but gel. Okay, this blue is nothing but gel. This is sample wells where DNA got loaded after excision endonucleases digestion, and here this is nothing but anode, and this is nothing but cathode. Cathode is represented by black wire, and anode is represented with the help of red wire, and this is nothing but direction of movement. Here buffer, the gel apparatus is filled with the help of buffer, and this is nothing but sample well, and here is negative charge, here is positive charge. It is positively charged, therefore it is called as anode, because anions, that is positively charged molecules, move towards the electrode which is mainly positively charged. Therefore the anions which are negatively charged move towards the electrode which is positively charged. Therefore gel act as a sieving effect, providing matrix. Therefore, they after loading the DNA inside this sample well, they run according to their size and shape. They got separated, and after separation, how smaller the size, more fast it can move, and higher the size, lower it can move with the help of milliampere current or voltage. So this is nothing but the image UV radiated image after staining with ethidium bromide. It is got it this the gel molecules or gel got exposed to UV light. After getting UV light, it got visualized inside gel dog instrument, and in that gel dog instrument, how it can get visualized like this. This is nothing but picture of gel dog instruments image, gel dog instruments image. So here this is 100 base pair, that is it's ATGC repeated 100 times. There ATGC is nothing but nucleotides. The DNA which is consist of 100 nucleotides got separated very early because its size is very less. 200 is more size, therefore it moves very slowly as compared to 100 base pair fragment DNA fragment. This is 200, this is 300, this is 500, 6, 7, 8, 9, 1000 and 1500 or 1200, and these are sample bills A, B, C, and this is nothing but ladder or a marker. With the help of marker, we can say that this fragment is nothing but 100 base pair, because here the uh, known base pair, we know uh, we know that the last will come 100 base pairs by segment, or it is a standard, and by comparing to the standard we can say that the sample BLC can contain 100 base pair fragment, B contain 200 base pair fragment, and here 750 base pair was conducted by B sample. So this is nothing but how we isolate the DNA. So here what we cut the DNA molecules or this gel, purify it and elute the DNA molecule, therefore called as the elution, and they got separated. Again, these DNA molecules got joined to its